Okay, thanks everybody for coming out on this rainy July day. Melanie is going to present her master's thesis to us today. Melanie is from New Orleans. She received her BS in Biological Sciences from the University of New Orleans in 2009. She's worked on several research projects across the United States, most recently on songbird stopover behavior. Is that right?
Stopover ecology.
Stopover ecology. Before coming to DSU, she's been an ECSC graduate student in my lab since January 2015. And next month, Melanie is going to take off for Curie Atoll in, well, tell us where it is.
It's a remote Hawaiian island in the middle of the Pacific.
She's going to be there for 6 months on a restoration project for 6 months. Exciting. I guess with no TV, right?
No TV, no internet, no phone.
You're lucky. Melanie's defense is the fourth from my lab, focusing on the importance of coastal barrier islands in Apalachicola Bay, Florida, for northbound migratory songbirds. When this research began 5 years ago, it was my goal to bring the global importance of this coastal resource into the spotlight. It wasn't on many folks' radar in Apalachicola region, just because traditionally studies have shown that more so northbound spring migratory birds depend more on the western side of the Gulf Coast, not the eastern side. But our research is showing that that's not necessarily the case. I think my students have done a great job in highlighting this coastal resource to managers in that region and to ornithologists really around the world. I mean, folks in Canada and South America have become interested in our research in Apalachicola Bay. So it's really an international interest that we've been able to draw to this resource. So Melanie's going to talk to us about this project. Again, this is the final students that I have that worked on this project. 3 came before her, Armando Escuro, Alan Neidl, Marimar Gutierrez. So Melanie's the fourth. Her title is Stopover Ecology and the Importance of 2 Apalachicola Bay Barrier Islands for Neotropical Migrant Songbirds During Spring Migration. So, Melanie?
Thank you. All right, thank you guys for coming. So here's a brief outline of my talk. I'm going to start off with an introduction, kind of giving an overview of barrier islands and neotropical migrant songbirds. And then I'm going to briefly discuss my objectives and hypotheses, and then talk about my methods, both in the field and in the lab. And then give you the most relevant results, brief discussion, concluding statements, and then hopefully have time for questions at the end. All right, so I thought I should start off talking about what is a barrier island and why we should care. So barrier islands are deposits of sand, gravel, and shell that were all formed during the Pleistocene or Holocene. And on the north coast of the Gulf of Mexico, they form physical barriers between oceanic ecosystems and inland wetlands on the coast. So they actually form a barrier that protects these inland bays, and so that leads to a lot of fishing industry in the area. They also protect coastal communities. So barrier islands take the brunt of tropical cyclones, and so they protect coastal communities from storm surge that occurs during hurricanes. So they're very important economic as well as ecological resources. Some of the ecological functions of barrier islands, they serve as nesting habitat for many species of sea turtles, threatened and endangered ones. They're nesting and wintering habitats for shorebirds, like the one you see here. But the area of my research is the functioning of them as stopover sites for migratory birds. Barrier islands are facing threats currently. So the whole world is experiencing sea level rise based on the expansion of water in the ocean and melting of ice in the poles. So because of sea level rise, barrier islands will experience migration, breaching, erosion, and saltwater intrusion. And this is on top of anthropogenic stressors that they are already experiencing, such as development and artificial stabilization. So these barrier islands are supposed to be naturally dynamic ecosystems, but once they're developed and artificially stabilized, they can't really change with the changing climate. And that's exacerbated by the rapid increase in sea levels. So the Intergovernmental Panel on Climate Change in 2013 predicted between 0.26 and 0.82 meters of sea level rise by 2100. But sea level rise isn't experienced equally in all places. So this is a graphic from United States Geological Survey, and the area of my research is in the Apalachicola Bay of Florida. And this report has given the Apalachicola Bay a high vulnerability ranking to sea level rise based on its high erosion rates and its low slope. So my research is focusing on neotropical migrant songbirds. So neotropical migrants are birds that breed in boreal and temperate forests in North America. And winter in Central or South America. So many of the species of neotropical migrants are facing declines in populations, and a lot of the research has pointed to the loss of habitat in breeding and wintering locations. But more recently, a lot of research has gone into the decline in habitat on the habitats that these birds use during migration. And this is on top of other anthropogenic stressors that they're currently facing. So anywhere between 500 million to 1 billion birds annually are killed by collisions with buildings, feral cats, and pollution. And that's in the United States. And then more recently, these birds will be affected by climate change and sea level rise. And so these factors will also contribute to the declining populations. So every year, birds migrate from the breeding grounds in North America down to South America and back. So this is a very energetically expensive migration that they make. It takes a lot of energy to move across the globe like this. But also, there's a lot of variability in migration between species. So every dot in this graphic represents basically an average for a species. So every species will take a different migratory route, and even individuals in a species will take different routes as well. So migration is very energetically expensive. Birds can spend approximately a third of their life cycle migrating, and it's the most vulnerable period in their life cycle. So this graphic is from a study with black-throated blue warblers. And this study estimated the survival probabilities of the species during the different times of its life cycle. And so during spring and fall migration, their survival probabilities are a lot lower than during the winter and the summer. So birds face a lot of threats during migration. They can come across inclement weather while they're migrating. They might not be able to find good habitat to refuel their fuel reserves. And this can make them more vulnerable to predation. So most birds don't make a direct flight from breeding grounds to wintering grounds. They do several short flights, and they use habitats in between to rest, refuel, and avoid predators. So these habitats are known as stopover locations. And stopover locations can vary in quality from one site to another. So they can have differing amounts of food, water, and shelter for an individual species. So for a lot of birds, the Gulf of Mexico is the largest ecological barrier that they'll have to cross. And if a bird is crossing the Gulf of Mexico, the first piece of land it's going to encounter after reaching the North Coast is a barrier island. So with a lot of birds, they will surpass barrier islands and go into coastal forests where they can find better habitat. But some birds aren't capable of doing this. So for example, if they encounter inclement weather during their crossing of the Gulf, this can create what's called fallout. And so when that happens, many birds, regardless of their physical condition, will have to stop because of either inclement weather or strong headwinds. And so a lot of birds will use barrier islands during fallout conditions. In addition to that, a lot of birds just arrive on the North Coast in physiologically stressed condition. So this could be a result of multiple factors. Maybe they weren't able to refuel properly on their last stopover. And so a lot of physiologically stressed birds will arrive on barrier islands as well. So barrier islands are important, especially important in those two circumstances. So for decades, researchers have known that coastal stopover sites are important for neotropical migrant songbirds. The majority of the studies in the last several decades have taken place on the north central or northwestern coasts. So a lot of studies in Louisiana, Mississippi, and Alabama. But in the literature, the northeastern Gulf Coast has not been recognized as important as those sites on the western Gulf. But more recently, this paper that I've taken this graphic from, they've used radar data where they can actually see birds migrating, and they've noticed this higher density of birds on the Florida Panhandle. And so they've hypothesized in this paper that this could be due to wind conditions over the Gulf Coast that can blow birds off course from either their Gulf crossing or the Caribbean crossing. So it's been recognized as a research priority to conserve high quality stopover sites. So how do we measure the quality of a stopover site? For years, people have been using this netting, so you can actually capture a bird and examine its physical condition. So you can look at the body condition, you can take measurements of mass or fat content on an individual bird. And these types of studies, recapture data can provide a lot of good data. So you can catch a bird, band it, release it, and then catch it a few days later and see how much mass it's gained or how much fat it's gained. And so that's really good, but recaptures don't happen too often, especially on barrier islands. One study found that only about 3% of birds are recaptured on barrier islands. So there's this other method where you look at where you regress the body mass of the species with the capture time. And with that, you only need a single capture from each individual, but you look at multiple individuals of the same species, and you see the overall trend in the species, whether it's gaining or losing mass. But more recently, a lot of research has focused on the use of plasma metabolites. So plasma metabolites are taken from a blood sample from a single capture event. So you don't need recaptures for this. Plasma triglycerides are a measure of refueling rate in birds. So this study here used captive Wilson's warblers, and they gave them a low-quality diet and then switched to a high-quality diet. And in as little as 10 minutes, they were able to see an increase in triglyceride levels. So triglyceride concentrations in the plasma are a measure of refueling rate pretty much in the moment that you catch the bird, or in the moments just prior to that. So the objectives of my study were to examine the use of 2 different barrier islands within the Apalachicola Bay, Florida, and examine the use by neotropical migrant songbirds. And I wanted to look at differences in capture rates, physical condition, and refueling rates between the 2 islands. And the end goal of this project was to provide data for managers to help them guide management decisions, especially as these barrier islands face increasing threats due to sea level rise, climate change, and other anthropogenic stressors. So these were some of the hypotheses for my study. I hypothesized that St. Vincent Island, which is the larger and less developed of the 2 islands, would provide a better stopover habitat for birds. So St. Vincent has a large variety of habitats, so I thought that birds on St. Vincent Island would be doing better during their stopovers. But I hypothesized that St. George Island would have more birds captured. And that's just because of the size and shape of St. George Island. It's much smaller, so birds would be more concentrated on I also hypothesized that males would arrive earlier, be in better physical condition, and have higher refueling rates than females. And that's based on other studies that have found that males arrive to the Gulf Coast earlier than females, and they also, in a lot of species, the males will arrive to their breeding grounds earlier as well. So I hypothesized that if they were under more stress to arrive earlier, that they would be refueling at higher rates and therefore be in better physical condition. I also hypothesized that the older birds, the after second year birds, would have higher refueling rates than second year birds, which are the younger ones, but they may not necessarily be in better physical condition. And this is based on a study of fall stopover in coastal Alabama that found that there was no difference between the age classes in terms of physical condition.
Hey, Melanie, hey, can you real quick explain the difference between after second year and second year to those that aren't ornithologists?
Yeah, so a second year bird is basically a bird in its second year of life. So when a bird is born, it's called a hatch year bird, and then after that calendar year passes, it's called a second year bird. But on the following year, that bird will be an after second year. And so when you're aging a bird, you can't really get it any more defined than after second year. So once a bird is 3 or 4 years old or 5, whatever, it's still called an after second year bird. So second year birds are experiencing their first spring migration.
Thanks.
And finally, I hypothesized that the time and date of capture might affect the refueling rate in birds and the ability to gain mass. Specifically, I expected that the time of day would affect them. With the increase in time of day, I expected to see an increase in body mass, and that's just due to them gaining mass throughout the day to prepare for their migration. So my research was conducted within the Apalachicola National Estuarine Research Reserve on the Florida Panhandle, and it's near the town of Apalachicola. The first study site was St. George Island. St. George Island is your typical barrier island. It's long, it's thin, it has a bridge that connects it to the mainland, so it's developed. There's a lot of residential areas on the island. There are a few forested areas as well. There's a state park on the eastern end of the island, and then there's Unit 4 where my research took place right there in the middle. And St. George Island is, or Unit 4 of St. George Island is managed by the Florida Department of Environmental Protection. And we chose a second study site, St. Vincent Island. St. Vincent Island is a national wildlife refuge managed by the U.S. Fish and Wildlife Service. It's a very atypical barrier island. It's triangular in shape. It has this well-developed dune and swale topography. And because of its topography, it's developed these different habitat types as you go through the island. So the elevation kind of increases and decreases. So you have a variety of habitats, including freshwater wetlands, pine woodlands, and oak scrub. And the banding station is marked right here with a pin. So for the rest of this presentation, I'm going to use the abbreviation SGI for St. George Island. So again, St. George is our typical barrier island. It's developed. This is a photo taken from Unit 4 where our research took place. Unit 4 has a canopy of slash pine, an understory of saw palmetto, oak, and wax myrtle. And as you get closer to the bay, it transforms to more of a tidal marsh with a lot of jumping species. In the habitat plan, they do have plans for prescribed burns, but there hasn't been a complete prescribed burn since 1999 in this area. St. Vincent Island, I'll use the abbreviation SVI for that. This is our atypical shaped island. It is undeveloped and uninhabited, but it does have roads that run through it for management purposes. So our banding station we set up on the intersection of 2 unused roads, so we're able to just line our mist nets right down the middle of it. And this banding station has it's a pine oak forest that borders a freshwater wetland. So we have a canopy of slash pine, a mid-story of oak and cabbage palm, and a thick brushy understory that you can see on the picture here. For the portion of my project with examining the blood samples and triglyceride concentrations, I used the gray catbird for that portion. So gray catbirds are the most abundant migrant in the Apalachicola region. And they're also a large size when compared to other migrants. So I was able to take a large enough blood sample that would allow me to do all the analyses on them. Gray catbirds are a really abundant species throughout North America. They breed in most of temperate North America, and they winter along the Gulf Coast, Central America, the Caribbean, and portions of the Atlantic coast. While I didn't take blood samples from the northern water thrush, they were my second most abundant species. So I ended up doing a lot of analysis with them as well. Northern water thrush breed through a lot of Canada, and they're only found in Florida during migration. And they winter in this area of South and Central America. An interesting thing about northern water thrush is that they are territorial during migration. So they'll actually seek out and defend territories where they can find good food resources. So a lot of birds don't really do that during migration. So for this study, we use mist nets to capture birds. And so this is a picture of a mist net that's set up, and they're supposed to be hard to see. So you basically spread this net between poles, and once it's opened, it's basically invisible. So the birds can't see them. They fly into the nets and get tangled. And then we come around every 10 minutes and extract the birds and bring them back to the banding table for processing. Once we have them at the banding table, we identify every bird to species, give it a uniquely numbered USGS aluminum leg band, and we take measurements like mass, wing chord, and tarsus. And all of that gets recorded on the data sheet. We also take measurements of fat and muscle scores. So birds store fat to use during their migration. They store it in the furcular hollow, which is right below their neck. So this bird up at the top, you can see this yellow portion under the skin. That's actually the fat that it's storing. So fat is scored on a scale of 0 to 7, muscles scored from 0 to 3. So this bird up at the top has a good amount of fat on it, and it's got these well-developed breast muscles as well. So that's a pretty healthy bird. In contrast, this bird at the bottom has used all of its fat reserves, and on top of that, it started to catabolize its muscle tissue as well. So this bird is physiologically stressed because it's used all of its energy reserves pretty much. I also collected blood from gray catbirds. So for every individual, I recorded the capture time as well as the time of blood sampling. I collected blood from the brachial vein. So that's the vein that runs down the wing of the bird. And while I was allowed to collect up to 10% of the blood volume, I rarely got that much blood. It was usually between 50 and 150 microliters. And so in the field, we stored blood samples in a cooler. And then when we got back to the field house, we centrifuged them to separate plasma from blood cells. The plasma was stored in its own separate vial on liquid nitrogen for the remainder of the field season. And the blood cells were stored with Queen's lysis buffer solution and kept at room temperature. I used the Cayman Chemical Triglyceride Colorimetric Assay Kit to determine triglyceride concentrations. So I first diluted each sample threefold and ran samples in duplicate or triplicate and then averaged the results. So this assay kit uses a spectrophotometer to measure the absorbance of each sample, and then from that you can use an equation to calculate the triglyceride concentration. Since our gray catbirds can't be identified to sex in the field, I used the residual red blood cells to do that in the lab. So with many species of birds, females have 2 sex chromosomes and males have 1. So I first extracted the DNA from my blood cells. I used these primers to amplify genes on the sex chromosomes, and then I finished with gel electrophoresis. So those samples with 2 bands, like the first one in the picture there, are females, and those with 1 band are males. For my statistical analyses, I used R version 3.2.3. I determined results to be significant when P was less than or equal to 0.05. So since my triglyceride samples were run in duplicate or triplicate, I determined a coefficient of variation for them. And if I received a coefficient of variation of less than 10%, I either reran the assay if I had enough plasma, or if I didn't have enough plasma, I threw out that sample for the remainder of my analysis. For many birds, the body mass is related to body size. So I adjusted the body mass to correlate with body size using the scaled mass model, which is described by P. Green in 2009. I used a chi-square test to look at differences between capture rates. And for the rest of my data, I ran a Shapiro-Wilk test. For the non-parametric data of fat scores and muscle scores, I used a Wilcoxon-Rank sum test. And for the body mass and triglyceride concentrations, I used t-tests. I used linear regressions for all of my temporal effects. And for triglyceride concentrations, since there was a lot of variables that I thought would contribute to that, I used a backwards stepwise regression to determine which variable or combination of variables were best at predicting triglyceride concentrations. All right, so on to the results. This is a brief overview of netting effort and birds captured. So one thing to point out here is that in 2016 on St. George Island, we had a crew from University of Massachusetts at Amherst was out there catching birds. So they worked on St. George Island and we worked on St. Vincent Island. So that's why we have the increase in effort and the increase in birds banded in 2016. And so they also shared data with us as well. So in both 2015 and 2016, St. George Island had significantly more neotropical migrants captured. Combining all of my neotropical migrant species, in 2015 I had higher fat scores on St. George Island, and but lower muscle scores on St. George Island. But in 2016 I did not have those same results. With my northern water thrush, I found no difference between islands when I looked at fat scores, muscle scores, or size-corrected body mass. And I've left out those bar charts just for, to ease with time here. But just to put this box plot in, just to show you the, like how similar the birds were between islands in both years. This is my regression between date and body mass with the northern water thrush in 2015. So St. George Island for the rest of these graphs will be in black, and St. Vincent will be in gray. So for both islands, I found a negative correlation between date and body mass in 2015, but it was highly significant for St. Vincent and not significant for St. George. In 2016, I didn't really find that same correlation. It was more of a positive correlation for St. Vincent, although it wasn't significant. This is my regression of body mass with time of day. And so for time, I standardized the time measurement by just recording everything as hours that passed since sunrise. So for both islands, we had a positive correlation, but it was not significant for either island. In 2016, we had a highly significant correlation on St. Vincent Island, but not for St. George Island. Moving on to the gray catbird. I found gray catbirds in similar conditions in 2015 between islands. In 2016, I had higher muscle scores on St. George Island. For my size-corrected body mass for the gray catbird, in 2015, I had a higher body mass on St. Vincent Island than St. George, but I did not find that same relationship in 2016. So again, I've regressed the body mass with the date of capture, and I did not find a significant relationship in 2015, although on St. Vincent Island it approaches significance with a negative correlation. In 2016, St. Vincent approached significance with a positive correlation, but I did not get any close to significance on St. George Island. So again, this is time of day in 2015, and I did not have any significant results here for either island. In 2016, I approached significance on St. Vincent Island, having a positive relationship between time of day and size-corrected body mass. So if a bird is increasing mass throughout the day, that generally points to that stopover site being pretty good quality. I found no effects of age on fat scores and muscle scores or size-corrected body mass in 2015. In 2016, I found no effects of age on fat scores or muscle scores. But in 2016, I found that second-year birds were, so the second-year birds are the younger birds, I found that they were significantly heavier after adjusting for body size. And I also found a difference in the capture date between second-year and after second-year birds, but only on St. George Island. So on the first year of the study in 2015, second years arrived approximately 5 days earlier than after second years. But in 2016, it was about 2 days after on average. So I was not successful with molecular sex determination in all of the samples that I tried. But I got about 72% success in 2015 and about 48% on St. Vincent Island in 2016. I did not attempt to sex the samples from St. George Island in 2016. But using the samples I did have, I found no effects of sex on fat scores, muscle scores, body mass, or capture date. In 2015, I found no difference between islands, sexes, or age classes in refueling rates. In 2016, I did find a difference between islands, but not between sexes or age classes. And these are my, my models for refueling rate. So this is a backward stepwise regression. So the way this works is on the first step, you run all the different variables and it gives you an AIC value. And then each variable is subtracted one at a time until you get the lowest AIC value possible. So for all birds, in this, all gray catbirds combined in the study, I found that year and fat were the variables that best describe the variation in triglyceride concentrations. And so I did a regression to see exactly what that relationship was. And as fat scores increase, so does the triglyceride concentrations. And that was the case for both years. In 2015, so I did the same analysis, but just for 2015 birds. And I found that location and date were the variables that best describe variation. And so again, I regressed it to see exactly what that relationship was. And on St. George Island, I had a significant relationship between date and triglyceride concentrations. So for 2016, I separated this out by islands because for one, I found a significant difference in the triglyceride concentrations between islands. But for another reason, the triglyceride assays were done by the University of Massachusetts crew. And so since it was done in a different lab by different researchers at a different time with a different assay kit, I just separated it out. So this is the one from St. George Island, and I found that bleed time and muscle scores were the best predictors of triglyceride concentrations. And so with increasing muscle scores, you get increasing triglycerides. And as expected, with an increase in bleed time, you get a decrease in triglycerides. And that's because triglycerides are very time sensitive. And on St. Vincent Island, the variable that best described the variation was age. But if you remember from earlier, I ran a t-test with age, and I got a p-value of 0.08. So it wasn't statistically significant, but it was still the best variable that predicted the variation. So my hypothesis that more birds would be captured per net hour on St. George Island was correct. So that doesn't necessarily mean that there's more birds on St. George Island. It's just because of the smaller area of forested habitat, all the birds that land on St. George are kind of funneled into a smaller area. And so the capture rates on St. George Island would be higher than on St. Vincent, where these birds experience a much larger variety of habitat and a larger area. So on St. Vincent Island, the birds are just more dispersed throughout the island. I was surprised to find that the northern water thrush was in very similar conditions on both islands. So I found no difference between fat scores, muscle scores, or body mass between islands. And I also found this positive correlation between body mass and capture time, which indicates that they are able to refuel and gain mass on the island. Northern water thrush are freshwater wetland species, so they forage near the water's edge and they pick up invertebrates. So they really require these freshwater wetlands to be able to refuel. And both of these islands had freshwater wetlands, but St. George is more developed and it's more fragmented. So I was very surprised to see that the northern water thrush seemed to be able to refuel appropriately on both islands. And another thing about them, I mentioned that they were territorial during migration. And so just being out there, while we were banding birds, we noticed some of the territorial displays of the northern water thrush, where they were, vocalizing at each other, chasing each other. So it seems to me that they were able to find and defend high quality habitats on both islands. With the gray cappard in 2015, I found gray cappards were on average 2.5 grams heavier on St. Vincent Island, but I found no difference in fat scores, and I found higher muscle scores on St. George Island in 2015. And when I looked at all neotropical migrants, I found higher fat scores and lower muscle scores on St. George Island. But when I had a larger sample size in 2016, I didn't find that same effect. And so I think what was happening is that on St. Vincent Island, we had a lot of breeding birds that would be classified as neotropical migrants, but some of them breed on St. Vincent and some of them travel further north to breed. And so it was very difficult to separate breeding individuals from transient ones. And so I think in 2015, we had higher muscle scores on St. Vincent Island because those breeding individuals don't really have a need to catabolize their muscle tissue. And again, lower muscles or lower fat scores on St. Vincent because those are breeding birds, a lot of them, and they wouldn't have the need to store fat. But with a larger sample size in 2016, I didn't find that relationship. So I think that that's what was happening there. So my hypothesis that St. Vincent Island would provide a better stopover habitat, my data doesn't really support this hypothesis. And so again, I think that was very surprising because Unit 4 on St. George is very fragmented. It's surrounded by residential developments, but it seems to be not too different from the National Wildlife Refuge that's right next door. But because of St. Vincent's larger size, there's obviously going to be more birds taking advantage of that area on St. Vincent. I found no significant effects of date on size-corrected body mass in gray catbirds, but it approached significance on St. Vincent Island. But interestingly, that was a negative correlation in 2015 and a positive correlation in 2016. So there's obviously different other factors at work here. So my study didn't go into other extrinsic factors such as weather or food resources. But those are definitely important things to consider with stopover ecology. So it could be that there's some outside variables at work here. But another interesting finding is that in 2016, I captured a single catbird that had been banded the previous year by us. And so that lends evidence to wintering site fidelity for the species. But it also makes me wonder if some of the birds that we were capturing were wintering residents and not necessarily transients. So that could have affected these correlations as well. I found no significant effects of time of capture for size-corrected body mass, although I did have a positive correlation that approached significance on St. Vincent Island. Two other studies have looked at this, have done the same sort of analysis with gray catbirds, and they've found differing results. In the study in 1992, they found mass loss with stopover sites in Minnesota. But in 1995, in the study in Mexico, they found that birds were able to gain mass during their stopovers. Another thing to point out is that I did have some smaller sample sizes. So for example, only 18 catbirds were captured on St. Vincent in 2015. So a lot of studies have suggested that this type of analysis with regressing size-corrected body mass with time of capture should only be completed with larger sample sizes, like anywhere from 30 to 120 individuals from what I read. So these results should be interpreted with caution because of that. And with my northern water thrush, I did find a significant negative correlation between body mass and date on St. Vincent Island. So I thought that that was probably due to females that would normally migrate later. And so, the later the date, the smaller the bird, because females are usually smaller. But I looked into that a little bit more, and there was a study published recently that said that you could sex some northern water thrush based on their wing measurements. And so just looking at that, I didn't really find a lot of definite females at the end of our season. And I did find some definite females at the beginning. So I don't really think that this correlation is because of differences in sexes. But it would be interesting to look at for a future study to actually get the blood sample to be able to sex the northern water thrush. And then I found a positive correlation between body mass and time on St. Vincent in 2016. And so again, this suggests adequate resources for the water thrush. And other studies have found similar results with an increasing mass throughout the day in New York City parks as well as banding stations throughout Canada. But again, this type of analysis is recommended for larger sample sizes. So again, results should be interpreted with caution. So my hypothesis that there would be no differences between the physical condition of second year versus after second year birds. My data supported that for fat and muscle spores, but not necessarily for size-corrected body mass. So I didn't find a difference between fat and muscle spores, but I did find that the younger birds were significantly heavier than the older birds. And they did find similar results to this during a study of fall stopover in Alabama, where they found no difference between the fat and muscle spores of different age groups, but they did find a difference in body mass. But I think that this could be related to to just smaller wing chords in the younger birds. And so I actually ran a t-test to see if that was the case, and I did get significantly smaller wing chords in the second year birds. And then another study to bring up is this study in 2003, looked at young birds in the first, couple weeks of their life as they're learning to become efficient foragers. And they did find that in the beginning they were kind of slow and not as good at foraging as the adults, but they quickly learned to become efficient. And by the time they were ready to migrate, there was no differences in the efficiency of their foraging between older and younger birds. And so it seems to me for gray catbirds, at least, that the younger birds are pretty efficient and can learn to be efficient very quickly. My hypothesis that males would arrive earlier, be in better physical condition, and have higher refueling rates was not supported by my data. I found no difference in arrival timing or capture time of males and females. But after researching this some more, I found that the study that looked at the breeding grounds on gray catbirds, they didn't really find that males arrived before females. So while this is the case in a lot of neotropical migrants, it doesn't seem to be the case with the gray catbird. So I think that male gray catbirds just don't experience the same time sensitivity as males of other species. And so this is probably why I didn't find what I was expecting to. And so I just put this little table here to sum up my best fit models for the triglyceride concentrations. And so I did find higher refueling rates on St. Vincent Island in 2016. But again, I can't rule out any effects of the different assay kit used by the University of Massachusetts crew, as well as the different lab, different researchers, things like that. So I can't rule out the effects of that. Other studies have found no effects of age and sex on refueling rates, although there are some species that they found higher refueling rates in males, but not all species. And my time of capture was unrelated to triglyceride concentrations, which I didn't expect to find either, but other studies have found that. And bleed time, which is the amount of time that's passed between capture and blood collection, that's been found to affect triglyceride levels in several studies. But we did have a careful protocol where we checked nets every 10 minutes and birds were bled as soon as possible. And if more than 20 minutes had passed, we didn't use that blood sample for triglyceride assays. But that was a relevant variable for the 2016 birds on St. George. All right, so just to summarize everything, while St. Vincent may not necessarily provide better habitat for neotropical migrant songbirds, it is larger and able to support a larger number of birds. So again, I pretty much found, I did not find differences between St. George and St. Vincent. And so I think they're equally important for neotropical migrant songbirds. A lot of data collected by birders have found that migrating birds often use small fragmented forests or woodlots or things like that. And I think this area on St. George Island is a small fragmented forest, but it's still providing good stopover habitat for migrants. And so I think a lot of small forests like that, especially when they're next to an ecological barrier, deserve protection for neotropical migrant songbirds. And again, there's been this idea that the conservation of stopover sites should focus on high quality stopover sites and not necessarily species specific stopover sites. And I think that that's the case for both of these islands, that they are high quality habitats and they seem to be working well for many species. So in the process of putting all this together, I kind of had a few more questions that I thought of that would be interesting to study. So a big one, I think, is that how weather patterns influence the ability to gain mass and refueling performance. Weather patterns are obviously a big deal for migrating songbirds, especially across the Gulf of Mexico. But I'm interested in how that would affect refueling performance. And additionally, the abundance of food. That's obviously going to affect refueling performance and the ability to gain mass as well. And I think it would be interesting to look at how those variables are going to change with the changing climate. So we will have different weather patterns in the future. And so how is that going to affect our migrating birds? While my sample sizes only allowed me to look in depth at 2 species, but it would be really interesting to look at other species as well if we could get a large enough sample size. So I think that these islands are important for other migrant songbirds, but it's definitely something that should be looked into. And I think that Apalachicola Bay barrier islands are very important for the northern water thrush. They seem to be gaining mass on both islands. But I think that more study needs to be put into their resource use and their territoriality, as well as refueling performance. So I wasn't able to take a blood sample from northern water thrush, but I think it would be interesting to look at refueling performance and how that's different between the sexes for them and different for more territorial birds and things like that. So I think there's a lot of interesting questions that we can come up with northern water thrush. And finally, I just wanted to put this back into the whole context of sea level rise and climate change. This is some research by Lori Lester, who is a postdoc in our lab. And in this graphic, the developed areas are in red and the forested areas are in green. And by 2100, the majority of the forest on St. George Island is gone, and it's mostly just developed areas. Of course, the birds are using the forested habitats, so depending on the severity of sea level rise, we could lose a lot of the forested area on St. George, and that would be very detrimental to the songbirds that are using the island. All right, and so that brings me to my acknowledgements. Funding for this project was provided by the National Oceanic and Atmospheric Administration's Environmental Cooperative Science Center. We also got a little bit of funding from St. Vincent National Wildlife Refuge with the help of Carol Brown, and that allowed us to expand to St. Vincent Island and conduct research there. I want to thank the Apalachicola National Estuarine Research Reserve, where our research took place. Specifically, Dr. Rebecca DeMangue, who was the research coordinator, and Megan Lamb, who works for the reserve and came out and helped collect data with us and helped with logistical support as well. I want to thank St. Vincent National Wildlife Refuge, especially Bradley Smith, who was very helpful with everything we needed out there. Delaware State University, my lab mates, Devin, Alan, and Armando, as well as Dr. Lori Lester, who helped out the first year. And in the Department of Biology, Dr. Tim Burney was super helpful with all of my lab work, as well as the students in his lab. And I want to thank the University of Massachusetts at Amherst, the Gerson Lab, and Maria Mar, who provided their data for me to use. And Maria Mar was also very helpful just in the beginning, just talking to me about collecting blood and about some of the lab work. I want to thank Florida A&M University, which is where the ECSC is housed, and my committee members, Dr. Heckscher, Dr. Mullinek, Dr. Timberning, and Dr. Barczewski. And I will leave you guys with these awesome pictures for any questions.
Questions for Melanie?
Yeah.
Well, I'll ask the obvious one. If it's so tough on them to migrate, why migrate?
Oh, that's a good question. I mean, there's a lot of different theories, but I guess it has to do with competition, with just moving out away from the competition between other species. That's a highly debated topic. So,
Is there any other reason that I mean, what you would think, there's plenty of insects and stuff down in the tropics. Why don't you just stay there? It's nice and warm all year round. Why come up to the colder climates?
yeah, I mean, I guess it could have to do with retreating glaciers back in the day. As these species were evolving, they were able to take advantage of more and more resources as glaciers retreated. So that could be what happened, just trying to get away from all the competition and exploring new resources.
It seems like some of your patterns might have been related to whether birds on the islands were migrants or actually residents. So obviously it was beyond the scope of your research to take that apart, but are there ways that people could, like, can people do stable isotopes for some unique moves or some islands to determine whether they're staying there or moving?
Yeah. I mean, if we were there long enough, we could have continued, banding birds through the remainder of the season, and then look at our recaptured birds and see, like, oh, we caught this one at the end of the season. We also caught it at the beginning, so it's obviously a breeding bird. That's definitely something that could have been done if we had more time to stay down there.
I haven't got anything snapshot wise that actually was possible.
Yeah, I'm not sure.
I mean, stable isotopes could get into that because, that will show you what the birds were, where the birds may have been feeding at the time that they molted, which would be on the breeding ground. So in other words, if you had a proprietary warbler that was in Delaware, it's going to show a different signature than a proprietary warbler on St. Vincent Island, right?
Right, but I don't think the signature from Apalachicola would be very much different from Georgia, let's say.
No, but you might be able to get at, some of the birds that may be going pretty far north, you might be able to get at that.
Yeah. So
the
that
other-
wouldn't rule them all out. But
The other question I was going to ask you related to that is, do you see any group patches on things like proprietary warbler? Because that would be an indication, right?
Yeah, definitely. Yeah, I think we did have some birds with brood patches. Yeah. So that's a possibility as well. Yes.
So if the sea level does rise and takes mid-forested areas and those two research areas, what's going to happen with the birds? Are they just going to find somewhere else, or do you think that they would just die off?
Well, so many birds don't depend on barrier islands. A lot of them are able to surpass them and go, further inland to more higher quality habitats. So the loss of barrier islands and the loss of the forested habitats would more affect birds during fallout conditions that I talked about, where birds are flying through bad weather or headwinds. Those birds might not be able to make it to the mainland. And then also birds that are just stressed out. So maybe they weren't able to adequately refuel on their last stopover site. So they've run out of fuel reserves. Those birds, again, might not be able to make it to the mainland. So for the majority of birds, it would be okay. But the weather patterns, I think, is a big concern because, that's going to change with the changing climate.
Could you, so one of the things I think that our lab has really brought to the forefront is the use of these islands by northern water thrush, which obviously your research shows that as well, and as part of that. Could you just very briefly talk about put it in a kind of a global or international perspective, maybe not just St. Vincent Island and not just St. George Island, but barrier islands on the Gulf Coast, how important they are maybe for this species. And I'm sort of asking that because I'm not really familiar with how common freshwater wetlands are on barrier islands in general. I mean, we have them on St. George and we have a lot of them in St. Vincent Island, but I don't know if other parts of the Gulf have barrier islands with a lot of freshwater wetlands, isolated freshwater wetlands. And you're from Louisiana, I don't know, have you been out to any barrier islands? So I'm just wondering if you could kind of put it all in perspective, just in a few sentences for folks that, managers in that region. I think one of the things that Carol Brown and the Friends of St. Vincent Island are really interested in is understanding how St. Vincent Island, how unique it is, if it is unique. They want to know that because she tells me they're constantly fighting to keep the refuge operating down there, and they need to come up with information for how important it is. And I think northern water thrush would be a great kind of flagship species for them. So, and I know the NER folks would be interested in that as well. So maybe you could just throw a sort of a global or international spotlight on the region.
Yeah, I mean, I'm not really certain how many barrier islands on the Gulf Coast have freshwater wetlands, but St. Vincent has a lot of them. And freshwater is important not just for the northern water thrush, but I mean, all migratory birds need fresh water. So yeah, I think one of the biggest threats is probably saltwater intrusion. I mean, that's going to happen in the lower elevation sites first. So I think the freshwater wetlands are very important for migrant songbirds, and they should try to be protected somehow from saltwater intrusion as much as possible. So St. Vincent's, it's a huge barrier island. It's very different from most barrier islands on the Gulf Coast. And just, I mean, even looking at the map, you can see some of the fresh water that's in there. Maybe. But yeah, I think St. Vincent is very important for the species, and there's probably, you know, we were only banding in this one little area, but yeah, there's tons of fresh water, and then even, you know, in each of these swales that you see in the ridges here, there's a little bit of fresh water in those as well. So St. Vincent is very different and very important.
Well, northern water thrush are species, are arboreal species.
Yes.
So where do they nest?
You
don't have to show us a map, but
yeah, they're in the boreal forests of Canada and a little bit of the United States as well. So yeah, I mean, that's a species that migrates globally. It goes from Canada all the way down to South and Central America. So it's interesting that we get a lot of these birds in Florida during a short period of time.
yeah, I'm sure other barrier islands get northern water thrush, but it could be, I mean, this is one of our most common species there.
Mm-hmm.
And it would be interesting to look at data from other islands to see if the capture rate of northern water thrush is higher for us on these two islands than elsewhere.
Yeah, definitely.
That's one of the things that we weren't anticipating, was having that species be one of the most abundant. And I'm doubting that on all barrier islands that's the case. So I think that for the northern water thrush, I think this Apalachicola region, these barrier islands are particularly important for that species.
Yeah.
Would you agree?
Yes, I would agree. Yeah, I mean, even just birding, I don't really see too many northern water thrush around. So it was really amazing to see how many of them were down there.
Since you have this map, these two islands seem to be right angles to each other, right?
Yeah, a little bit.
So the direction in which the birds come, would that be a big, and then you mentioned wind directions.
Yeah, so it's all very variable with wind directions. But I mean, in general, the birds would be coming from the south probably, although it depends on the wind. You know, if you have strong west winds, they may be coming from further east, and vice versa. So it's very dependent on weather. But we did try to get our banding station, you know, pretty far south on the island to take advantage of that.
Any other questions for Melanie? So, alluding back to one of the questions I was answering, or asked a few moments ago, if we lost all of St. Vincent Island and all of St. George Island to sea level rise, I'm going to try to get you to say something. What do you think the impact globally would be to these birds? Or would there be an impact?
Yeah, I think there
I mean,
would
should someone sitting in an office in like Florida or Texas or wherever, should they be concerned about losing these two islands?
Yeah, I think these two islands are globally important resources. As I mentioned in the beginning, these neotropical migrants are already facing dramatic declines in many species. And so I think without these islands, that would just be exacerbated further for many species, in particular the northern water thrush.
Well, the other obvious thing too is that if these islands go, other islands go. So
Right,
it's not just
exactly.
these two islands. It's a
Yeah.
lot of area.
And I mean, it's in parts of the coast as well. So the coastline may not be where it is now. It's gonna be retreating further back. So it's gonna be an even further distance for these birds to migrate.
Yeah, I mean, I put it on, I mean, it's been suggested to me that barrier islands aren't that important because, you know, if you think of the global population of these bird species, it's only a small percentage that might be stopping on these islands, which I think is kind of debatable, but nevertheless, even if that's the case, I think it deserves as much concern as feral cats, which has become a major concern. Building strikes, I mean, you said early on in your presentation, how many birds did you say are killed every year?
Estimated between 500 million to 1 billion annually in the United States alone. And that's just from collisions with buildings and cat predation.
Yeah.
And pollution, I think.
My opinion is that it might be a little bit high, but obviously it's a major hit every year they take. And I would throw this into the pot with things like feral cats and building strikes.
Yeah, definitely. Yes.
Is there any problem with feral hogs then?
They do have feral hogs on St. Vincent. They've been trying really hard to get rid of them. I'm not sure. I mean, I don't think that directly affects the birds, but definitely affects the habitat for sure. You can see the damage that they cause by their rooting all over the island. But they've got a guy out there who's actively trapping the hogs, and he's doing a really good job. So hopefully he keeps it up.
Sure, barbecue there.
Yeah. Yes.
One thing I'm questioning is, like, you know, barrier islands are somewhat like an ephemeral piece of land, you know, they shift very rapidly. So is there like any sort of record they could see, like when birds actually started using these islands as like a stopover habitat, like when they actually, I know it's a little out of question, but I was kind of curious.
I mean, I would assume that they always have. I mean, it's very, the use of these islands is very dependent on weather. So, I mean, pretty much any time a bird is migrating with bad weather, it's gonna need these islands.
Okay.
So I'm pretty sure they've always been using them.
It wouldn't have been there. in the first place.
That's true.
Yeah, that's
But
right.
there may have been other islands.
Well, there was the Florida Ridge, which was in itself an island.
Yeah.
And so that may have served as,
But
as the
yeah,
stop.
of course there may have been something else.
Right, but Florida was pretty much inundated at that time, so that would probably include these barrier islands.
So it's like a fully terrestrial migration at that point, I think they just completely braved the Gulf and so forth.
What about Cuba? As a stopover site, 'cause that's kind of in between.
Yeah, I'm sure a lot of birds use Cuba for sure. The thing with this area is that a lot of birds are taking off from the Yucatan Peninsula. So some birds do go around and use Cuba and Florida, but a lot of birds just cross straight over from the Yucatan.
Yeah, Cuba is a major stopover site. Yeah, it's at across the Caribbean Sea and all that.
There were people doing research there, so it's pretty hard to get permits to go there.
There's a lot of Cuban scientists that are working there, so.
I'm sure they gave us a go for it.
It's actually more than you would believe,
Really?
yeah. It's just that we don't hear about it.
Any other questions for Melanie? All right, thanks, Melanie.